Hello everyone, welcome to today's session. Today we are going to see Class 12 Biology, Chapter 4, Principles of Inheritance and Variation. In this we are going to see inheritance of one gene, that is the monohybrid cross. So inheritance, what is inheritance? Inheritance is a process by which traits are passed from parents to offspring. To understand how a single trait is inherited, Gregor Mendel, he performed experiments on pea plants. So this monohybrid cross led to the discovery of the basic principles of inheritance. Yes, let's start with the topic. Monohybrid cross. So monohybrid cross is a cross involving one pair of contrasting traits. So Mendel here, he selected the plant height in pea plants because it showed two distinct forms. I'll show you here. So what he took the tall plant and the dwarf plant. That is with a he crossed a true breeding tall plant. Yes, this is a true breeding tall plant and this is the true breeding dwarf plant to study the inheritance of a single gene. So after crossing the two plants, he collected the seeds and grew them to produce the first filial generation. So first filial generation, what happened in first filial generation? All plants were tall. All plants were tall and none were dwarf. So the F1 generation resembled only one parent. Then what he did, he allowed the F1 plants to self-pollinated. Yeah, to self-pollinate. F1 plants to self-pollinate. So in the second filial generation, he found the dwarf character reappeared. So here you can see the dwarf character reappear. So among the offspring, 3/4 were tall and 1/4 were dwarf. So it gives a phenotypic ratio of 3:1. So what is the important thing you should see here? See, an important observation was that the offsprings were either tall or dwarf. But there were no plans of intermediate height, right? So it was tall or is dwarf. So no intermediate height was there. So it proves that characters do not blend during the inheritance. So what it proves? Characters do not blend during inheritance. So Mendel, he obtained similar results for all other contrasting characters, whatever he studied. So one parental character appear in the F1 generation while both characters reappeared in the F2 generation. So what you should remember for the exam point of view is monohybrid cross. Yes, monohybrid cross studies one character and F2 phenotypic ratio is 3:1. So no blending of characters occurs here. Making a cross in P. So first what he did artificial cross pollination. Why he took this phenomenon? Because in order to ensure that only the selected parents produced offsprings, and Mendel he carried out the controlled pollination. So what are the steps in controlled pollination? First is emasculation. So in the step 1 he did the emasculation. What he did, the flower, the selected flower, which is like a female parent, had its anthers removed before maturity. So here you can able to see here in the picture, yes, the anthers are removed before maturity. Why? So this prevents the self-pollination because pollens cannot be produced by that flower. That's why. And next one is the pollination. So next in the pollination, what he did, uh, so pollen grains from the selected male parent. So this is the selected male parent. So carefully transferred to the stigma. Carefully transferred to the stigma of the emasculated flower. So in the next step, so after fertilization, you know what will happen, right? We already saw in our previous videos. So when this pollen grains are transferred to the stigma, pollen tube will be formed. Here it reaches the ovule part, and then what happens? Fertilization process takes place. So after fertilization, the ovary develops into a fruit containing hybrid seeds, so which produces the next generation. So what you should remember from this is, see, controlled pollination only ensures that the observed traits are inherited only from the selected parents. So exam tip is here. So emasculation is nothing but the removal of anthers to prevent self-pollination. So next we are going to see genes, alleles and symbols. So Mendel's factors are genes. Yes, Mendel's factors are genes. Here it is. So from his observation, he concluded that something was passed like from unchanged from parents to the offspring through gametes. So he called these heredity units as factors. He called this as the factors. And today we call that factors as genes. So genes are the units of inheritance. Why it is called as the units of inheritance? Because they carry the information needed to express a particular character. So different forms of the same genes are called as alleles. So different forms of same genes are called as alleles. So these are alleles like, yeah. So for a plant height, so this is the tall, yeah, this is the dwarf one. So the capital letter is used for the trait expressed in the F1 generation, and this a lowercase letter, it represents the recessive trait. So what are the possible genotypes we get? We get genotypes as TT, yes, this capital T, small t, two small t's, right? So true breeding plants, it possesses identical alleles and they are called as homozygous. So here identical alleles you can able to see these are homozygous, and here also identical alleles, so they are also homozygous. But this plant, this one have different alleles, so they are called as heterozygous. Okay, so exam point of view you should remember genes control the characters while alleles are alternative forms of the same gene. Next, we are going to see genotype, phenotype, and dominance. So, genotype and phenotype. See, genotype is the genetic constitution of an organism. For example, we can say, see, this capital T, capital T's, capital T, small t, small t, small t. So, these are the genotype. And phenotype is the external appearance, how they look. Is it a tall plant? Is it a dwarf plant? Yeah, the external look of the plant. And Mendel, he observed that the heterozygous plant is TT, the heterozygous plant TT, it looked exactly like a capital T, capital T. It looked exactly like capital T, capital T. So he concluded that one allele can mask the expression of another allele. Yeah. So what he said, one allele can mask the expression of another allele. So he said that T is dominant, capital T is dominant and small t is recessive. So this phenomenon he called as the dominance. So although two capital T's and capital T and small t, they are genetically different, but both appear tall. Yes, children, both appear tall. So phenotype alone cannot determine the genotype. So what you should remember is always please use this T and this T. Yes, and you should not use T and D for dwarf. Okay, children. So this is wrong. Don't use because alleles of the same gene should be represented by the same letter. Next, we are going to see the Punnett Square. Before, see, understanding this Punnett Square, who developed Punnett Square? See, it was developed by Reginald C. Punnett. Who developed it? Reginald C. Punnett. So this is a graphical method actually. This is a graphical method. It is used to predict all possible offsprings produced in a genetic cross. So graphically you can able to see this method. So in the cross, what happened? First he took TT. Can you able to see? I'll show you here in the picture. So capital T, and he took a here small t. So the two gametes produced here are tall parent, that is the capital T, and a dwarf parent small t. So all F1 plants, it like became tall because you can able to see here T is dominant, capital T dominant, capital T dominant, capital T dominant. So all the F1 are tall only. Then again now he self-pollinated this F1 plants. You can able to see, yes, capital T, small t, capital T is self-pollinated. Again he crossed, and what he found? So each parent now, see, it produces two kinds of gamete. It is having both like a tall as well as the recessive. So what happens? So each gamete, it has a equal chance of carrying either allele. That is 50% chance was there. So equal chance of carrying either allele because the alleles separate during meiosis. Then random fertilization occurred and it produced, you can able to see capital T, capital T, capital T, small t, capital T, small t, small t, small t. So what is the genotypic ratio here? So here one capital T, one capital T, and here 1, 2. Yes, so 2 here, and here small t, 1. So the ratio is 1 is to 2 is to 1. Genotypic ratio is 1 is to 2 is to 1. Here you can able to see. Then phenotypic ratio. How many? Here capital T is dominant, capital T dominant, capital T dominant. So 3. So here you can able to see 3 tall and 1 dwarf plant. Yes. So the phenotypic ratio differs because T is dominant over small t. So what is the exam point of view? You should remember Punnett Square predicts the both genotypes and phenotypes. Let's move on to the next one. Yeah, let's see the test cross. So what is the purpose of the test cross? A tall plant may be capital T or capital T small t, and they both look identical. But the phenotype alone cannot identify genotype. So in order to determine the genotype, Mendel crossed the unknown tall plant with a homozygous recessive plant. And this is called a test cross. Yes, and he found if all the offsprings were dominant, the unknown plant is capital T, capital T. If offspring appear in a like a one tall, one dwarf ratio, the unknown plant is capital T, small t. Yes, that is only given here. And let's see one example here, children. So this is there in your book. So what he has taken? He has taken like a homozygous recessive. So this is the homozygous recessive plant. And this is also a homozygous recessive. So two small W's are there. Here also two small W's are there. Then he crossed this with the a dominant phenotype. See here the dominant phenotype can be both two capital W or one capital W and one small w, right? So he crossed it with the this phenotype, dominant phenotype. And what we saw here, see, we have to draw one table like this. Yes, these two alleles we are going to write here, and these two capital here capital W, and we are going to write this. So this is what is this? This is dominant. Yes, this is dominant. So this is dominant. This is dominant. Yes. So here what happened? All flowers are violet here. So here the unknown flower is a homozygous dominant. Hence proved here. Yes, unknown flower is a homozygous dominant. But here what happened? Here also we did the same process. Here it is dominant, dominant. But here both are not a dominant. It's a recessive, right? So what happened here? Here two are violet flowers and two are white flowers. So half of the flowers are violet and half of the flowers are white. So unknown flower is a heterozygous. Yes, here it is proved, right? So it is a heterozygous. So definition for the test cross. So a test cross is the cross between an organism. It shows a dominant phenotype and a homozygous recessive organism to determine its genotype, right? Let's move on to the next one. Next, we are going to see the Mendel's law of inheritance. So, Mendel's law of inheritance, two laws are there. One is the law of dominance and another one is the law of segregation. Let's see what is there in the law of dominance. Law of dominance is the first law. So based on the monohybrid process, Mendel he proposed that characters are controlled by discrete units called genes. And first it was named as factors, then now it is called as the genes. So genes occur only in pairs, that we know very well, right? So genes are, so these are the genes, it occurs in pairs only, right? Yes. So they are occurring in pairs. So in a pair of unlike alleles, one allele is dominant. Yes, in a pair of unlike alleles, one allele is dominant and other is recessive. It is proved. So this law explains why one parental trait appears in F1 generation and why in F2 phenotypic ratio it is 3:1. Yes, next let's see the law of segregation. This is the second law. Here, the alleles of a gene, they remain separate and they do not blend. Already we saw in our previous slides also. See, one tall character is formed or the dwarfness is shown. No intermediate characters are expressed there, right? So during gamete formation, that is the meiosis, the two alleles separate. So each gamete receives only one allele. Each gamete receives only one allele. A homozygous parent produces one type of gamete. Yes, a homozygous parent produces one type of gamete. And a heterozygous produces two kinds of gamete in equal proportion. So this law is also called as law of purity of gametes. Yeah, let's see the incomplete dominance. So in incomplete dominance, neither allele completely dominates the other. So as a result, what happens? The heterozygote it shows an intermediate phenotype. We already saw in our previous slides. See, tall will be there, dwarf will be there, and no intermediate was formed in the F1 generation. But here, an intermediate phenotype is occurring. Yeah, let's have one example. Snapdragon. Snapdragon is commonly called as the dog flower or antirrhinum. Yeah. So let's see this one. So we have taken the red flower, so which is having both the dominant R, capital R, and a white flower which is having both the recessive, that is small r and small r. Let us cross it. After crossing, we were able to find a pink flower. Yeah, is it red in color? No. Is it white in color? No. It is intermediate. It is pink in color. Then again, it is self-pollinated. So both selfing is done. Then in the F2 generation, we were able to find one red, two pink and one white. And also how the genotypic ratio was is capital R, capital R, and small r. So here the phenotypic ratio is equal to the genotypic ratio, that is the 1:2:1. Yes, children. So here the dominance depends on the function of the gene product. Yes, children, always bear this in mind. So next let's see the codominance and multiple alleles. So in the codominance, both allele express equally in the heterozygote. Let's take this ABO blood group. So the I gene, yes, it has the three alleles. The I gene here it has the three alleles. You can able to see here three alleles. So the I gene has three alleles. It is IA, IB, and a small I. So it is how we are saying it. See, IA and IB, they produce the different sugars on the surface of the RBC. Different sugars on the surface of the RBC, nothing but the red blood cells. And the allele I, that is a small I, it produces no sugar. So when IA and IB occur together, both sugars are produced. Yes, when they occur together, both sugars are produced. And if only A is there, A is produced. If B is there, B is produced. If no sugars are there, O group. Okay, children. So, what we can prove here that IA and IB are codominant and both are dominant over I. Next, let's see the multiple alleles. In multiple alleles, let's take again this ABO blood group. It demonstrates the multiple alleles because the gene has 3 alleles in the population. Yeah, we know, no? So what are the 3 alleles? We know that is the IA, IB and the small I, right? So what happens here? Every individual, it possesses only 2 alleles, one inherited from each parent. So the codominance is both allele expressed together. Codominant means both allele expressed together, and multiple alleles means more than 2 alleles exist in a population. Yeah, let's see a quick revision, children. So monohybrid cross studies the inheritance of one character, and genes are the units of inheritance, and alleles are different forms of the same genes. And tall T is dominant over dwarf small t, and F1 generation shows only the dominant character, and F2 phenotypic ratio is 3:1, and F2 genotypic ratio is 1:2:1. And alleles separate during meiosis, that is the law of segregation, and test cross, it determines the genotype. And incomplete dominance, yes, incomplete dominance, it gives 1:2:1 phenotypic ratio, and codominance is illustrated by AB blood group, and ABO blood group also demonstrate the multiple alleles. So we have come to the conclusion part. Yes, watch the full video, don't skip it. Yes, it will be very useful for you. We will see you in the next video. Thank you.